it can make this portion carry out the work. And this particle never had to enter the cell. So they can act as points of attachment. So now, wow, got quite a few proteins going on. Transport. When substances actually get to move, whether that would be movement from extracellular to intracellular or intracellular to extracellular. Hold up. We keep talking about all this stuff that would need to get into the cell, but I just told you about a movement that could be inside the cell to outside. Any idea what could move from inside the cell to outside the cell? Hormones. Do it, Patrick? Waste. All right, I'm going to get you guys to think about something else. Well, hopefully, viruses and that sort of thing are
Broken attached to the entire cell.
they can work in two ways, okay? Viruses can either take over the DNA of the cell, or they can go into the cell and reproduce themselves. So there's two ways viruses can work.
Maybe like more frozen.
Yes. Our cells of our body have to make their products. The only product they can make is a protein, which is coded for by the gene on the DNA of the nucleus. So, for example, insulin. We're all familiar with that, right? Okay. What organ produces them? Our pancreas. So, not only do the cells of the pancreas have to be doing all this maintenance, repair, reproduction, so forth, they still have to make their product. Their product is insulin. So, this is an example of their movement of a material outside the cell. Does that make sense? So not only do cells have to worry about getting stuff into themselves, they have to make sure materials move out. So we have transport proteins. Transport proteins, based on the structure of the protein, we're going to see that we have a hydrophilic region, hydrophobic region, once again of the proteins, based on how they're embedded in that plasma membrane. And because of that, these transport proteins can help determine what molecules can pass. Now, we have proteins that act as a channel. I kind of think about a waterway when I hear channel, okay? And a channel is just where, you know, the ships just get to move back and forth. There's really no control other than making sure you're not going to hit each other, okay? So with the channel protein, the materials just simply get to go back and forth. All right? Some, however, will have a gate. But we'll talk about that. But just one that's a channel, it's just letting stuff go back and forth, back and forth. Some can be a carrier protein, meaning whatever gets to move has to actually fit the channel of that protein. If the shape of that molecule doesn't fit, it doesn't get to move. Then we have ones that need ATP. They need energy. For them to work, ATP needs to give energy to that protein. For the most part, cells like simple movement. But in some cases, they have to expend energy. They have to use energy to get materials they need. Channel proteins. Now here is where things get interesting. In that channel protein, all right, if it's just there, no gate, no control, all right, if a substance can fit into the channel, that substance gets to simply move back and forth. If the cell needs it, it'll use it. If not, it just simply goes back across, and it'll just wait for the next movement. Those are non-gated. There is no gate anywhere controlling movement. They're very important to homeostasis of the cell. But we have proteins that are channels, but they're gated. What do you think of when you hear something has a gate? Restricted. So these proteins that can exist, they're channels, but now they have a restriction. Something has to make them open. Meaning, if the gate is based on being a ligand, do you know what a ligand or what that term means? To ligase?
Ligand is just like a small particle. It could be a variety of things.
And if we think about what occurs in the process of a ligand or to ligase something, Tori? Well, yeah, because these are proteins. These are proteins with channels. But if something needs to ligase, it needs to connect. So, a ligand is a particle. Okay? So, out here in my extracellular fluid, I have a ton of particles. Okay? I mean, we have neurotransmitters. We have all these nutrients that the cell might need. We've got all these products that are in the extracellular fluid. So I've got a lot of particles. So on this protein that is a channel, but it has a gate, but it's based on having a ligand being present. That means it's going to have somewhere that a particle can fit, that it can bring together. So, if there's a particle out here and it attaches to the area that's available and it fits, what do you think is going to happen to the gate?
It's like a lock and key.
Huh?
Said it's like a lock and key.
The connection between the protein and the ligand, yeah, it has to fit. So that means it doesn't just accept any particle that's out there. The particle has to fit into the site that's available. But when that happens, what does the gate do?
Open.
It opens. This gate will open. Does that make sense? These two go together, the ligand and the voltage gated, because for the most part, in this situation right here, when a ligand gate opens, it's perfect, the size is perfect to allow ions to cross. Sodium ions are very plentiful on the outside of the cell. Now remember, the inside of the cell is mostly negative. A little bit of positive, but mostly negative. So now, if sodium is very plentiful, and this gate opened, and it's the perfect size for sodium to cross, sodiums flow inside. What did I do to the charge to the inside of my cell?
Balances out.
Nicole?
Did you make it more positive?
We're making it more positive. So now, all these little negatives become positive. I think this one's back on. We're now seeing that I get a rush of the positives inside the cell. So I now take that negative and I've now turned it positive. Does that make sense? And I have now affected, AFF, okay, affected my membrane potential. That's the thing that was mentioned earlier. Take a break, be back at 9:30.